Warm greetings to my dear students. In earlier session we discussed about the specific properties of enzyme, and now we'll talk about factors affecting enzyme action. So we'll be covering important factors that influence the velocity of enzyme reaction. First of all, we'll talk about effect of temperature. So how can we say that enzyme activity is related to temperature? So scientists earlier, they designed an experiment by keeping all the factors constant and only temperature was kept variable, and temperature was increased gradually. So they plotted a graph between enzyme velocity and temperature. Enzyme velocity against temperature. So the velocity of enzyme reaction increases with increase in temperature up to maximum and then declines gradually. So a bell-shaped curve was obtained. With this experiment, they concluded two things. First is that there is a range in which enzyme works, and second, that there is an optimum temperature at which enzyme activity is highest. So with this, we get to know that velocity of enzyme reaction increases with increase in temperature, and optimum temperature is what? When rate of reaction is maximum. Remember that enzyme is most active at optimum conditions. So now suitable temperature for enzyme action is 20 degrees to 35 degrees. Most of the enzymes are active in this specific range. However, there are some enzymes which work at extreme temperatures. And with increase in every 10 degrees Celsius, there's 2.5 time increase in rate of enzyme activity. And at high temperatures, denaturation of proteins takes place and enzyme activity becomes slow and finally stops. So what is denaturation? Denaturation means change in the structure of proteins. Denaturation takes place at 50 degrees Celsius or above 50 degrees Celsius. Now we'll come to effect of pH. That means hydrogen ion concentration. So all the enzymes are sensitive to pH of the medium, and each enzyme has an optimum pH at which velocity is maximum. Below and above that pH, enzyme activity is much lower, and at extreme pH, the enzyme becomes totally inactive. So most of the enzyme work efficiently between 5 to 7.5 pH. However, there are some exceptions where optimum pH is more than this. So a graph was plotted, enzyme velocity against pH. So the enzyme velocity increases as pH increases. But at certain limit, the enzyme velocity stops and starts declining gradually. So a bell-shaped curve was obtained. Now we know that enzyme velocity increases with increase in pH, and rate of reaction is maximum at optimum pH. Next is concentration of substrate. Let us understand with the help of a graph. Enzyme velocity against substrate concentration. So on increasing concentration of substrate, enzyme activity also increases. But when concentration of substrate is increased beyond a certain limit, then the rate of reaction becomes stable. So we get A part which is linear, and B part which is a curve, and then C part which is a constant. And at this point we achieve Vmax. That is the highest rate of reaction at optimum concentration of substrate. That is Vmax. This is optimum concentration of substrate. So concentration of substrate can be minimum, optimum or maximum. At minimum concentration, enzyme activity is low due to no combination of substrate on all the active sites. But when concentration of substrate is increased beyond to a certain limit, then the rate of reaction becomes stable. Now comes concentration of enzyme. So let's look at the graph. As concentration of enzyme increases, velocity of reaction also increases proportionately. But on increasing concentration of enzyme, the activity gradually increases, but due to substrate limitations, because substrate is limited, so at some point of time it stops and becomes constant. Now comes concentration of end product. Now, when reactions are taking place, the end product is formed, and when there is accumulation of end products, it decreases the enzyme velocity. So this decrease will reach to a stage of equilibrium where the activity becomes zero and reaction may proceed towards reversible direction. Next is enzyme inhibitors and poisons. So what are enzyme inhibitors? Substances which reduce the catalytic activity of enzymes are called enzyme inhibitors, and they react with enzyme activity and makes it non-functional. They can be organic or inorganic in nature, and further they are divided into competitive inhibitors and non-competitive inhibitors. Let's look at the diagram. See, this is the enzyme molecule and this is the active site of enzyme. And this is substrate in blue color and this is competitive inhibitor in pink color. And this is the enzyme inhibitor complex. So, in competitive inhibition, you can observe the inhibitor resembles substrate. The structure resemblance you can observe. The competitive inhibitor, it resembles the structure of substrate. So, the competitive inhibitor occupies the active site and forms enzyme inhibitor complex. But in case of non-competitive inhibition, see, this is the enzyme molecule, this is the active site, and this is the other site where the non-competitive inhibitor will bind. This is substrate and this is non-competitive inhibitor. See, there is no structural resemblance between these two. And the non-competitive inhibitor, it will bind at the other site and forms enzyme inhibitor complex. And it will bring structural changes in the active site of enzyme molecule. See, earlier it was this and now it brings the structural changes in enzyme molecule. So there are some differences between these two. First of all, there is structural resemblance in competitive inhibition and there is no resemblance in non-competitive inhibition between substrate and non-competitive inhibitor. Second difference, the inhibitor competes with substrate to combine at active site of enzyme molecule, because of which enzyme activity decreases. And in non-competitive inhibition, the inhibitor permanently attaches with enzyme molecule and brings about structural changes in the enzyme molecule. And in this case, inhibitor binds directly to the active site, of the enzyme. And in this case, the inhibitor binds to the other site and not the active site. And this, in competitive inhibition, it can be overcome by increase in concentration of substrate, and in non-competitive inhibition, it can be overcome by increasing concentration of enzyme. That's it for now. We have done all the factors affecting enzyme action, and in next lecture we'll start with mode of enzyme action. Thank you so much.